now. We have 34, 35. There's still quite a few people joining, so just wait another minute or so here for a few more. And then we'll get underway. Let's see now. Oh, the names went in alphabetical order, so I can't shout out more countries. That's fine. All right, well, maybe we'll get underway now. So welcome to this month's Speaking of Oats webinar. My name's Charlene White, and I work for Agriculture and Agri-Food Canada. I'm also the editor of the Oat newsletter, which is why I'm here with you today. With me is Jim Bradeen. Jim is the director of Oat Global. He's also a professor and department head at the University of Minnesota. And we also have with us Grant Chadzek, who's with the University of Minnesota Communications team. And before I introduce the speakers, there are a few housekeeping items that we need to go over. So first off, as you've already seen when you joined, this webinar is being recorded. It will be posted to YouTube, and the links will be made available through both the Oat Global website and also the Oat newsletter site. And at some point, I'll post the links to those sites in the chat. Speaking of chat, there are 3 ways that people can connect with the panelists and everybody else online. So the first one is to use the chat box. So that is at the bottom of your screen. Now, chat is meant for chat. So any side discussions, further discussion, also any comments and so on, please put those items in the chat. For actual questions, there's a Q&A box there as well. So please put your actual questions in the Q&A box. Now, some of those may be answered during the seminar, or we may leave them all to the end. We'll just see how all of that flows. The third way you can communicate with everyone is to request to be added as a panelist, in which case we can unmute you, we could see you on video and so on, and you can actually speak to the group. So if you feel you have something to add where you need a bigger platform, as it were, then you may ask to be added, or we may actually ask somebody to join the panel as we did last time. So those are the 3 ways. And so I guess, yeah, that's it for the housekeeping stuff. So our speakers today are Taner Shen and Vic Blake. Actually, Victoria Blake, sorry, a little more formal for this. And Taner is a lead scientist with GrainGenes at USDA-ARS in Albany, California. And Vic is a longtime curator at GrainGenes. And she's now, though, at Montana State University in Bozeman, Montana. So Vic and Taner, I will pass the floor over to you now.
Thank you, Charlene. Okay, let me get started. So first of all, I'd like to thank the Oat Global and the Oat newsletter for inviting us to the presentation. So I'll talk in the first 10-15 minutes about the GrainGenes content and our stakeholders, and then Victoria will take over and then talk about the markers and QTL and CMAP, showing how geneticists can use GrainGenes for their research. So there is no mystery. GrainGenes is a centralized repository for wheat, barley, rye, and oat. And so this is our website. This is our link. We are supported by USDA-ARS and also US Wheat and Barley Skip Initiative. And we are very grateful. And I don't, although I am we are like me and Victoria are presenting this. This is a work of decades, and so currently we have a mixture of curators, geneticists, and programmers working together to present this resource to the community. We are also overseen by the GrainGenes Liaison Committee. This is a committee of experts of International Committee of Small Grains, geneticists, and breeders. It's a combination of wheat, barley, and oat researchers. And we meet once a year, and they tell us what we are doing right and what we need to be working on in the future. So we are grateful for their time. that volunteer time for our database. As I said, the GrainGenes mission arose from the need of centralizing the data sets that are generated. So these are genetic data sets, genomic data sets, phenotypic data sets. So it's a large range of data types. And our stakeholders are global small grains researchers. So if you look at our user base, it's actually largest in North America, Europe, India, and China, but it is also other countries as well. And we have been around since 1992, so we do have lots of data, especially genetic data. We don't do this as an island. We actually collaborate extensively. One of our main collaborators is our Nick Tinker and Charlene White that help us with the oat data curation into GrainGenes. So that's they're from AFC. We're part of the Pan Oat Consortium. So Martin Mascher from IPK and Nick Tinker are leading this. And I think you heard in this series about the Pan Oat Consortium a couple of months ago. GrainGenes is also part of the Oat Grant Challenge. And Jose Costa next month will be presenting about the Oat Grant Challenge that USDA people actually are getting together to answer questions, to create data, and then to answer questions related to nutrition and other areas. We recently collaborated with PepsiCo for the release of OT 3098, and this was PepsiCo's way of creating data and sharing it publicly with globally through GrainGenes. And we are an ARS database, and we collaborate very closely with 30C Toolbox, but also with other databases as well, like MazeGDB. And we are part of this AgBio group that brings together different databases of agriculture-related databases from academia as well as ARS. We also have other collaborations, especially when it comes to wheat. As you know, it's one of the top staple crops, and also international wheat information systems. But I won't talk about these today. So let me just show you what we have. So if you go to the GrainGenes website, this is a very practical way of looking at GrainGenes. So you can look at the search and browse GrainGenes on the left-hand side. That's actually, let me start from the right. So if you go to the website, if you want to see what is new at GrainGenes, then you can look at the GrainGenes updates on the right-hand side. So you can look at the month by month, what is new, and you can see the new data sets, and then you can find the data you like. You can follow us through social media. We have a Twitter account that we post our updates. We post new positions and conference information. We do have hot topics in the middle. So these are sometimes it's a very big announcements like 10+ wheat genomes, or in this case, PepsiCo exploit oat genome assembly release, or sometimes just very interesting papers that our curators chose. So let me just go from the left-hand side now. So this is the search box, and you can actually search or browse GrainGenes. So if you click on that, you will go to the search side. So on top, you can actually do a text-based search based on using Google, or you can choose different classes of data sets, and you can look at the records specifically in that way. So the first part is that it doesn't look at any classification of data. It just looks at text. So the second part is more specific. You can also in the search area looking at the genetic maps at GrainGenes, and there's a complete list of genetic maps here. So it is divided based on species. There are wheat and barley, and of course oat maps divided by hexaploid and diploid species. So if you go to the bottom of the GrainGenes homepage, then there are upcoming events. Here you can see different conferences, different meetings. We have species portals. We have annual newsletter, Barley Boulevard, Barley Genetics newsletter, but also Oat newsletter is here as well. So if you click on it, you go to the Oat newsletter that Charlene White is editing. And we only provide a place for the Oat newsletter to exist. So we just help with the infrastructure. So it is hosted by GrainGenes, but we don't have any other involvement in terms of the content. So if you go to the community services part, we have calendars and current hot topics if you want to look at the other hot topics. But we also have data download that the people can use. So there are two aspects of it. When you click on it, you go to the GrainGenes data downloads, and there are specific data download pages associated with the data that we have. But also, you can go to the GrainGenes tools and advanced queries. And here, if you're inclined to it, you can use our SQL interface to get data from our database. We also have mailing lists. One of them is a GrainGenes mailing list. Another one is Oatmail mailing list that we manage. That Charlene White is actually also the editor for that. And you can just go there, put your name and email address, and then you will part of this mailing list. And this is a curated set, curated mailing list. We only send emails regarding to conferences and job listings. If someone sometimes asks questions about small grains, like what can I do? How can I find this type of questions? But this is not geared towards usually for undergraduates. These are for more like postdoc and professor level or PhD students. Job listings in the community services is an area that people visit. Of course, I guess. So if you click on it, you can see that there are different levels of jobs. It can be postdoctoral research associate positions. It can be graduate research assistant position, or more research leader, center director. So it is all kinds of positions that we post here that are related to small grains. If you go to the community services tutorials, then we have the video tutorials. These are also available on YouTube, and you can learn how to perform simple custom SQL queries, for example, in GrainGenes, and different ways of finding how to find data at GrainGenes. If you go to the top of the menu item at the GrainGenes tools, you will have access to the type of tools that we actually host at GrainGenes. So we have, for example, genome-specific primers. Recently, we put RJ primers. It's an older program, but we made access to it so that you can reach it from the front page. Batch primer tree and other tools like CMAP that Victoria will talk about, and then BLAST and genome browsers. Actually, you can find some of those tools in the quick links. So under genome browsers, for example, here, and BLAST and CMAP. So this is the general overview, and specific Oat resources and tools at GrainGenes are genome browsers, the BLAST, genetic maps and markers, and Oat newsletter hosted by GrainGenes. So if you look at the genome browsers, we do have shared genome browsers with the 30C toolbox. So the 30C toolbox is another ARS database. If you guys were in the last month's webinar, Jean-Luc Yannick presented the 30C toolbox. And we do have very complementary missions. They are dealing with raw data, providing tools to researchers, and we deal with the peer-reviewed data. So we decided to have common shared genome browsers to save for efficient use of resources. So going forward, actually, every browser we have is in collaboration with the 30C toolbox. So specific Oat genome browsers we have, and remember that the Pan Oat project is ongoing, and next year we'll have more genome browsers, more, maybe 20, 30 more. So we have PepsiCo, we have diploid oats, and hexaploid oat from 2013. So what's a genome browser? So this is a view of genome browser. So very basically, we do have some information about that genome browser. In this case, it's PepsiCo, and you can actually see where the data is located or who you need to contact for more information. So on the side, on the left-hand side, we do have the tracks. So these are the representation of the nucleotide sequences as one-dimensional structure. So in this case, we do have the pseudomolecule itself, and we do have the genomic features and genomic markers that are aligned to that genome assembly. So you can choose those, and then on the right in the middle of it, you can see the genome features aligned to the genome assemblies. So it's a very easy way of seeing what is available, what is around the region that you're interested in. So you can also go from one genome browser to another using the genome drop-down menu on top. If you want to download something from the genome browser, you can do that. For example, for this track, you can learn more about this track, from using the About This Track option, or you can save track data and see those, get those coordinates or different track information, multiple track information at the same time as a downloadable file. This also shows how we linked between GrainGenes and other databases. For example, if you press on this marker, you'll have more information about it, like their attributes. But also, we do have a link to T3, and when you press it, you will go to the T3 site. So the other way is also available. If you go to T3 site, so T3 takes trial information from different collaborators, and using automated GWAS tool, they create the plots, and then from that, through meta-analysis, they obtain measures, cumulative evidence for different loci. So then, if you go to the then in the T3, for example, if you look at the GWAS results and put, for example, grain yield, then you will obtain a table. And from that table, if you see, you will have some values, but also links to different browsers. So in this case, you can use the GrainGenes link to go to the GrainGenes genome browser for that region. So we have that cross-linking between databases because the important thing for us is to serve the users. And if the user can actually go from one database to another easily, then this will help them in their research. We also have BLAST. So we have some old collections, as I mentioned. So then as a user, you can put your sequence of interest and choose the old collections that you like to blast against. And then you obtain a result space like this. And if the old collection has also a JBrowse link, then you can press on the JBrowse link to see that region on the genome browser. So we are linking not only between different databases, but also between different tools to facilitate knowledge discovery. So with that, this was very fast. So I'll give the microphone to Victoria.
Hey, thanks, Tanner. Okay, so I'll start sharing. All righty. Okay, can you see my screen? Okay, terrific. Well, thanks, Tanner. He gave a really nice introduction of our project And like Charlene said, I've been bouncing around the database community for quite a while, and I've been back working with GrainGenes full-time for the past 3 years. And in those 3 years, I've spent a lot of time working with Charlene White and trying to get our oat collections current. And she's done an amazing job. So most of what I'm going to be showing you today, she should get half credit for sure. What I'll be doing today is taking you through the oat-related content, some of it, and a short demonstration on using CMAP. All righty. So last year we had the mandate that we really needed to enhance our germplasm collection and increase the links out, make it reciprocally linked to T3 and GRIN, which I'll show you a picture of GRIN. I'm sure most of us are familiar with that. In that endeavor, I selected about 300 lines from T3 that were either part of a breeding panel, part of the oat core collection, and all of the germplasm lines that were used to construct the Pepsi genome map. There are a little over 300 of those. And from those, I enhanced them whenever possible with the pedigrees and links to the URLs for T3 and for GRIN. So I should note really quick before we go on. So at T3, they really do have all of the versions of Canoda that you'll find. It was an old line. It was first released in the 1929. So there's been a lot of reselections since then. In GrainGenes, we really just try to keep the original name and not like we don't want to, it's not going to be confusing, but again, with Canoda, we mostly just link to different references, polymorphisms, a lot of, again, the original data. With Canoda, you can see we have 26 genetic oat maps that are connected to this particular line. Yesterday, as I was going through a lot of the previews of this, I found there's a PVP list from 2019 at the Practical Farmers of Iowa. So that is something that's going to be a nice thing to add. I think if there's plant variety protection on these lines, I think it should be reflected in the database. So I'll be working on that in the future. Okay, so clicking on the URLs, for T3, I use Canoda mapping. You can use this link to go to GRIN. That's the Germplasm Resource Information Network, USDA. It has kind of a new look. This is really the front page. So if you go to the Canoda line, this was the original accession. Again, 1929. Some states have restrictions. You can order it from GRIN. And then up here, all these links will have different things you can click on and get more information about this. But this isn't a GRIN talk, but I just wanted to point that out for those of you who haven't looked at GRIN lately. So we also link to T3. These are just a few screenshots scrolling down from the same page. I selected the Canoda pipe mapping. So at T3, you could have a lot of versions of Canoda. And if so, they separate what distinguishes this particular line with either the PI mapping. There's a little pipe there. So scrolling down, there's more information about that, all the different names. In theory, there's got to be some Canoda there that links back to GrainGenes, because I know I sent all of those links back. So we should be linking back and forth. This particular version does have experimental data attached to it. So I included that. And then again, the Purdy pedigree you saw last week is a nice interactive way to show the pedigrees. At GrainGenes, we have pedigrees, but they're in a string, a Purdy string generally. Okay, so more types of information at GrainGenes. We have genes, as you might imagine from the name of our database and our project. Since we work on wheat, barley, oat, and rye, and other things, when we put a gene in, you won't see this like this published, but we do put the genus name with the gene. So you can distinguish, a user can distinguish, say, 6-PGD from Avena from the triticum version. So if you want to see all of the genes right now, you can search with Avena in parentheses. Another thing you can do is if you wanted to search by just the string, like PC for the crown rust resistance genes. We've been working on that quite a bit, and there's more work to do because PC exists both with the dashed and undashed versions. So one of the things I've been trying to do is work very hard to make sure that there are synonyms there, because if you see them both ways in the literature, you're going to search that way. So I don't want people to come in to get no data if it's there. So we're not about to argue what taxonomy is right. We're just trying to accommodate everybody. I won't spend a lot of time on this, but GrainGenes has been a repository for genetic markers for decades now. And if you think about the evolution, I mean, we've been doing mapping for a long time, but it started out as just phenotype, right? Milling yield, for example, it's really slow and it's very expensive. You have to grow all your populations out and then test them first. So researchers and breeders have been really, improving our ability to use molecular markers like map-assisted selection and be able to really test those populations out when they're very young, sometimes even in the seed, to see what genes you have or what markers you've carried over from the plants you're interested in. So I'll just go through quickly genotypes. We do have isozymes and GrainGenes. A lot of the original RFLPs came out of Oats, and these were just taking random cDNAs. And then you can find polymorphisms. We didn't always know what they did. We just knew that there was a messenger RNA attached to it. It was pretty expensive. It was slow. It used up a lot of radioactivity. So it was good to move on from there. The next thing that came out, AFLPs and rapids. These were faster. They were cheaper, but they weren't really targeted either. And some labs I heard had a lot of difficulty repeating the experiments, but we do have a lot of maps with these types of markers. Simple sequence repeats, labs started going towards that, made a lot of progress. And those are they're also called microsatellites. And they're places in the genome where you can get a lot of repeats, like maybe 3 codon repeats over and over. And since those are very polymorphic, that's a very good thing to use to map genes or area, not even genes, I think it'd be just areas of a genome. Diverse array technologies came out with a new technology. It's very good, but it's proprietary. So while we do have DARTs in GrainGenes, the marker information is very limited. But I try to, with every marker that's a DART marker, I try to put a link over to diversity arrays technology, so you can write to them and get those markers if you wish. And finally, lastly, SNPs. Sequencing is getting really cheap. And so we're able now to really produce a lot of SNPs really quickly. So there's probably at least 100,000 of the AV, Avena, GBS, genotype by sequencing SNPs in GrainGenes. Okay, another thing we work with a lot are QTL. So I'm sure most of us are familiar with what that is, but often when you're doing mapping, you don't really know what gene is causing your trait, but you can map it to a region. It's sort of an early step in identifying this. So right here, here's a QTL for example, beta-glucan for kernel weight. So we don't know what gene in here it is, but we can pretty much narrow it down to the region. So again, if you're doing crossing and you know what markers are in here, you can look at your progeny and say, okay, I probably have that bit before growing out the entire population. And then we try, when we're curating QTL, to put as much accessory information as we can. We link to the ontology group when we can. We try to include as many synonyms, significant markers, peak heights, different statistics that are available if possible. Okay, so Tanner mentioned maps. This is one thing that GrainGenes has been working pretty hard with. And so the genome browser that he showed you is kind of like a physical map. And you can really think of this as like a slide rule, where every bit of it is equally distant from the next because it's all molecular. It's like one long molecule. And then this is just a cytogenetic map. This is sort of a cartoon. Genetic maps are different because they really are, you make a map by showing linkages. So depending on where on the molecule your 2 loci might be, there might be more or less ability to make crosses there when these are basically making their grandchildren. It's kind of, I don't know, I want to get into that. But a genetic map might not always be synonymous exactly with a physical map. I guess that's what I'm trying to say. All right. We have a lot of maps in GrainGenes. And again, this is something that Charlene has really been helping me with. If you click on the genetic maps, you will show this complete list like Tanner showed us before. But then if you want to say, go down to the oat maps, the hexaploids, diploids are separate, then you just click that and it will take you there down on the page. For GrainGenes users, I want to let you know, we're working right now on improving this page a little bit. and to try to have more information. But what I do with this is I include a link into GrainGenes, and then I try to put the year, the parents, how many markers, what types of markers, and how many linkage groups. They're in order by year. So if it's a good place to go if you want to see if your parents that you're interested are been mapped or anything like that. Another good way is just start from the reference. That's a pretty good way too, if you don't know how we're going to map those genetic maps. We have over 100 now, and again, most of them are due to Charlene White's really diligent work last week. We share a template. It's an Excel template, and so she'll take the references, fill out the template, send it to me, and then from that I can easily make an actual GrainGenes map record from it. So big shout out to her. So next I want to show you a little demo. I'm hoping I have enough time to do that. I'm going to go through some parts of it pretty quickly. But what we're going to do is we're going to show how to use the large 2018 consensus map to improve to enrich the markers around PC53. That was just published a few years ago. Okay, so once you have a gene record, there's not really a link from a gene into maps. You have to look for a locus record first. So PC53 from this particular paper was mapped in 2 populations, and this one in the center is centrally mapped. This one was up on one of the telomeres, so let's use that one first. So this is what it looks like when it renders in GrainGenes. So clicking on that map link will render the image. Oh, sorry, this came from you didn't see the top-down approach. This kind of just got copied out of a tutorial I just did. That should be on YouTube any minute. Oh gosh. So yeah, by clicking on that, we're going to try to improve the area around PC53. So you can go down, you'll click Add Maps. What that will do is it will bring up a map reference set for us. Oh, I'm sorry, I'm trying to go through these, so it's, okay, let me just play that through. So what you do is you go down and you click Add Maps, and that's going to populate a table that has any map set with corresponding markers. It doesn't show you, every map set in GrainGenes. It filters them, okay? So what we can do is from here, you can see that these are all the OAT maps that have corresponding markers. The OAT 2018 consensus map has 2 linkage groups, and Merge 8 has 23 corresponding markers. So we're going to select that, click the Add Map, and then it draws it. But what you can see is PC53 was mapped inverted when compared to the consensus map. So there's a way of fixing that. And I hope this can scroll down. Okay, so you can scroll down, you can click Flipped there. And you can also redraw. Okay, and then there's another way of you can highlight PC53, click redraw, align, and then it will highlight that. So if you go down, highlight features, and this is really, this isn't going to change your map, but say you want to show this on a poster, it'll save you a little time in PowerPoint, right? To have CMap actually highlighting this for you. All right, so now you have this aligned, but okay, well, we can get a little better, right? So if there's a way of exploring the CMAP details, I'm not going to take us through that, but this particular linkage group has 4,660 loci, and we're only seeing, a handful of them, maybe 40 or 50 here. So in doing that, you can say, all right, this is mapping somewhere between sort of 70 and 90 centimorgans. So we're going to scroll down. I hope I can get this, hope this works for us. You scroll down. Okay, let me just take us through this quickly. Once you get this big enough and you get this zoomed in enough, one can see that it really maps in between 79 and 81. So I went back in and I changed those boundaries again to 79, 81. I hit redraw. And now you can see that here's this one little snip that co-localized with PC53. It's actually in this large linkage group that's co-localizing. So one can get information about all of these markers and go back and maybe do a little more fine mapping of the map you're interested in, and potentially even find what gene it really is. So you can click on that. It's going to, once you go there, it's going to, you can click on any of these markers on a consensus map. Once you click on it, you'll get a feature page. From there, it's going to cross-reference into GrainGenes. And then it'll take you to a locus page. From there, you can, oh shoot, it didn't play for us, I'm sorry. But from there, well, it'll take you back into the GrainGenes page, take you into the locus page. From there, you go to probe, and from probe, you go to sequence, and then that will show you where the SNP is. So sorry about that. I was trying to make this go a little faster, but I ended up kind of messing up the animations. I should tell you though, if you're interested in seeing this tutorial as it was intended and slow and methodical, you could just visit the GrainGenes official YouTube page, and this tutorial should be available there today. And it's called Using CMAP to Enrich Markers Around a Gene of Interest. So thanks everyone for your attention.
Great, thank you. Really informative presentations. And we are waiting for questions. Just a reminder to put them in the Q&A box for our panelists. I'm curious off the bat, of course, this is a genomics focused tool. Can we access transcription data through GrainGenes? RNA-seq data or?
No, not really, not yet. Ensemble has been a better place for that, to be honest, for transcription type data. Yeah, it'd be nice, but I mean, it potentially, you know, there's certainly room for tracks on the genome browsers once we have those types of data. In the SQL data, not in the SQL database, not so much. Tanner, do you know if any transcription? Transcriptome data is?
Right. So for the raw data sets, the reads, they need to go to the NCBI database. We only have the transcripts if they are processed. So for example, if somebody did an analysis on the expression levels of the genes, so we can put those type of data as tracks, but not the raw reads. I guess there's another question about, is it possible to download the flanking sequences of SNP markers from the GrainGenes T3 database? Yes. So essentially you go to the SNP marker that you're interested in, you then, you can zoom in or zoom out to the region, and then you just download the track information the way I showed it. So it's like there's a track info, you just click on the right-hand side and then it says save track. You can do it, you can download the sequences that way.
Mm-hmm. There's a tutorial about that too. So if you check our tutorials page, there's one that gives a little more long slogged out directions on how to do it.
Sure.
Great. Another question just came into the Q&A box. Thank you. I have a question not related to oats. In the past, the mapping population was developed from Steptoe times Morex to study the malting aspects of barley. I was wondering if it's possible to access data such as malt extract related to these mapping population lines.
That's something you'd look at T3 for, to be honest, because T3 has more of the, the raw data. And if it gets, let's say someone gets all this raw data and they put it together and then they can map the QTL for malt extract, that's when you just kind of see it in the GrainGenes. But that sort of supplementary raw data, generally we don't have that. T3 is really the place to go for that.
Great. We have another question, this one for Victoria. I've been working on matching CMAP together with physical maps, which are based in base pairs, by myself. Does the GrainGenes database provide these kinds of consensus maps for both centimorgans and base pair?
We do put the maps in CMAP as physical maps if we have those data. There are a few of them now. There are two of the diploid maps, Ariantha. Oh God, I don't remember the name of that one. Sorry?
Atlantica.
Thank you.
Atlantica, yeah.
Yes, and we have those as browsers, and then Nick Tinker put together a few of the subset of those maps where he was able to match up the SNPs that were genetically mapped and those physical maps. So we have a few of them in CMAP. It would be good to have more for sure. And I think once, the problem with CMAP is you can't really accommodate, okay, let me step back. A lot of times when you do physical mapping, it might be GBS markers, or it might be these unique marker names. And if you put those all in CMAP and there's nothing else out there in genetic maps that have a match, it's kind of pointless, right? So until we really start seeing like the HORVUs for example, for barley, we're starting to see publications now where they're saying, okay, here's this whole gene series of ADH, for example. ADH1 is really HORVU this, blah, that. Now we start having the opportunity of actually creating physical maps that you can have working with CMAP and then really use them comparatively. I don't know, and I think in a more efficient way. Certainly, please write to us if there's anything you think that will help you in your work. Let us know. We certainly take suggestions first.
Absolutely.
There is a couple comments from Nick that are relevant here too. These are in the chat box for anyone who's interested. PepsiCo is releasing version 2 of OT 3098, and we will soon be adding a GBS track for SNPs. Soon, he promises. And this will enable CMAP comparisons as we have for the diploids now.
Good.
And once V2 is released, we will create a genome browser and data download and BLAST tools for that.
Right.
Great.
While we're waiting for more people, I actually can't ask a question as a panelist, as it turns out, but I suppose this is really more of a request than a question anyway. So thanks, Vic, for the shout out. Yes, I have been doing a lot of work with Vic, and it's been really great working with her and the GrainGenes team. So thank you, Vic, for all of that. And as she mentioned, there are a lot of oat maps now that we have done and that are available on GrainGenes, but there's still quite a few missing. And so, for example, I have about another 20 in my queue of maps to be done that I'll be sending along. But I'm sure I've missed a lot of them. So if people go through the list and notice, hey, you're missing this work, you're missing this work, please send links to the work either to Vic or to me so that we can actually get those maps curated and added to the list in GrainGenes.
Yeah, we did not mention specifically the oat mapathon that Victoria and Charlene did at GrainGenes. I don't know, so many maps.
Yeah. Yeah, certainly oat is the best represented species with respect to genetic maps that we have on GrainGenes now. And that's Charlene's doing, makes it easy.
Thank you.
Well, I'm being selfish because I wanted to look at genes in QTL. So
oh, no, that's good.
It helps immensely. Well, once everything's pulled together in a database, I mean, it makes a huge difference. It's so much easier to mine the data. So having things like GrainGenes and all the tools and T3 as well, and so on and so forth, is just wonderful for those of us trying to answer, biological questions and so on. So thanks so much for all of that.
Sure.
Thank you.
I should make a comment too, though. We did touch briefly on SQL queries. There's a lot of times when we don't have a pre-made query for something in GrainGenes, but if you write to us saying, I need to pull out all the information with this and this and this and this, write to us. We can probably write a query for you. And I'll send it to you and you can use it again in the future. And I'll show you how to use it. So.
And the way the best contact us is to go to GrainGenes website and the right-hand side on top, it says feedback. Just press that button and then there will be a box there with your name, your comment, and we will answer you right away.
One additional comment from Nick in the chat. It was Charlene's brainstorm to say that we can put a map in GrainGenes even if there are only 2 markers involved. So.
Yeah, that actually came about because one of the things, because I mean, I've been doing mapping for many years too. And so one of the things that always kind of bothered me was there's a huge history of literature with classical genetics maps. So particularly to do with things like rust resistance and rust resistance genes. So that literature was always very separate from the, the more molecular marker mapping work. And it suddenly occurred to me one day that, well, you know, if you go back to some of these old papers, they say, well, you know, this gene is tightly linked to this other gene. And it's like, oh, well, that's a map. You have these two genes and they're linked at 0 centimorgans. So can we put that sort of information into GrainGenes? And actually it was quite a challenge for Vic, I must say, because CMAP and the GrainGenes, the way it's set out, really doesn't like that. However, if you have two things at 0 distance, but Vic solved that issue by adding a flanking marker, just called flanking marker. So if you find some of that really old information, sometimes you just have two genes, say PG3 and PG9, for example, together at 0 centimorgans, and then a little ways down, it'll say flanking marker. And so that's just there really as a placeholder to make it work.
Yeah.
But it does work. And then of course, when you pull that up, then you can link that to all the other information in the database. So, yeah.
Just to point out that I think almost all, maybe all our maps have more than 2 markers.
There are quite a few that have only 2 markers. Yeah, well, again, because of all this classical genetics work, there was an awful lot of that done. And most of it, again, having to do with the rust resistance genes, but also things like onnedness, lemma color, various traits like that. There's a lot of literature from sort of, I think the oldest paper might even be back in the late 1920s. So before people even knew what DNA really was, people were finding these types of linkages, and that information is important. And so that's how, you know, because we know now that rust genes do cluster in certain particular locations in the genome, and now we can visualize that more easily through all of these tools. So.
Great. Any additional questions from our audience? Or any closing comments from our panelists?
I'm looking forward to hear Nick again so that I can ask lots of questions. Well,
we definitely appreciate your time and insights today. So thank you both very much for being part of this.
Thank you.
Thank you.
Charlene, do you have any additional comments to offer?
Oh, well, Grant has actually, Grant, you just sent that to the panelists. He's added a few more links there that people might like to have. No, I just would like to thank our speakers very, very much. So thank you, Tenor. Thank you, Victoria, for a great job. And next time, we will have a whole slew of people. In fact, Tenor gets to speak again next time, because we will have a number of scientists from the USDA-ARS all working on oats, and they will be speaking. Jose Costa is going to be overseeing that group of people. And we do actually have a slide. Grant, could you put up that other slide, please? Oh, and there is actually one more question.
Yes, a late breaking question just came in. So let's get to the question first. So the question is anyone identifying the new rust race from 2019 and 2021? I don't know if the panelists have insights or maybe somebody in the audience.
Yeah, I don't know. Are you speaking, this is from Carlos, are you speaking specifically of a new race that's being found in the field or a particular gene? If there's someone in the audience who has an answer, perhaps you could raise your hand or put something in the chat or the Q&A. Otherwise, we can follow up afterwards. I don't see anybody volunteering.
Could make a great topic for a future speaking of oats.
Right. We'll have to see if we can find it. Oh, wait. Oh. Oh dear. So yeah, so Carlos is saying that it's a new and aggressive leaf race occurring 2019 and 2020. I'm assuming that's in Brazil. Perhaps maybe Carlos would like a minute to speak. Can we allow Carlos to speak?
Yeah, Grant, would you?
Okay, I think Carlos, if you can unmute yourself and turn on your camera if you want to, you can give us a little bit more information. You're still muted. There we are. Hello.
Well, good afternoon. Yes, we are having here in, I think in Brazil, maybe in some countries in South America, a very aggressive race of the leaf rust, these 2 last seasons. And we don't see any work or anything trying to identify this new race, if it's a known race or whatever. If you have any information, it will be very important for us.
Yeah. So you know that it's completely new. It's overcome some of the
Overcomes
genes?
many of our varieties here in Brazil,
Okay.
or most of it, I think.
Yeah, we can put that, I can put that request in the newsletter update and ask for people to respond. Sure. Yeah, okay. Well, I'll put that request in the newsletter because I don't think right now we have anyone who can answer your question. Great.
But thank you for making us aware.
For sure. Okay. So I think we were at the point, unless there were other questions and so on, Grant was about to put up the slide for next month's Speaking of Oats webinar. Also, Grant, yeah, Grant is the, so is registration open?
Yes, I put the registration in the chat.
Right, so you did. Wonderful. So if there's nothing else for now, I guess we can finish off. Thank you so much again to our speakers, Tanner Shen and Victoria Blake. It was wonderful to have you. And we very much look forward to seeing our whole crew from the USDA and Tanner again next time. Thank you also to all the attendees and people who've interacted. Very happy to see everybody. And yeah, I guess we'll see you next time. So thanks very much for coming, everybody.
Thank you.
Thanks.
Thank you, everyone.
Bye.